Good evening, and I will be teaching you today how to stain a cheek cell. But before we get into the how-to part of this procedure, we want to figure out what it necessarily means to stain a cheek cell. Why would we want to stain a cheek cell? Now, underneath a microscope, it can be semi-difficult in order to identify the cells. So when we stain or dye the cells, they can be much more easily deciphered. Now, the materials that we would want to use for this process are toothpick. It doesn't make sense now, but it will later. Glass slides, cover slips, paper towels, water, methylene blue, which is our dye, and then a microscope. Now the steps for this would be step number 1, place a drop of water onto a slide and then set that to the side. And then step number 2, we want to collect our specimens, aka our cheek cells. So in order to collect our cheek cells, we want to take the toothpick and then we want to lightly scratch the inside of our cheek. And then we go on to step number 3, which is to place the specimens into the water. And how we do that is we just take the edge of the toothpick and then rub it gently into the water. Step number 4, we're going to apply the dye, which is called methylene blue. So we just want to take one single drop of this and then place it in the water and leave it for a minute, and that should be enough time in order to completely dye the cells. Step number 5, we want to cover the slide with a cover slip, and make sure that you're doing this at an angle as to avoid air bubbles within the space between the cover slip and the actual slide. And then we want to eliminate the excess water, which we can do with the paper towels. Now, if you can put drops of water on the edges of the cover slip, and then once you do that, you can see the water underneath the cover slide will start to retreat away from the drop of water, and that's when you take paper towels and then lightly press it against the edge of the cover slip, and then it'll start to extract the water. It should, when you're finally done with it, it should come out either clear or with a light blue tint. And then step number 6, we want to place our slide underneath the stage clips, turn on the light, and adjust the diaphragm just to make sure that we have the correct amount of light. And then step number 7, we want to focus on low and then focus then. And then we can switch over to medium, focus there, and then we can switch over to high and then focus there, and then that is the point where we can start to see the cells. And then once we've finished these processes, voila, we've completely stained a cheek cell and we're now able to easily identify them. Thank you for watching.